Good morning, everyone. Thank you so much for joining us on a bright and early Sunday morning. No better time to be here to talk about our arch nemesis, guinea worm disease. It is my distinct pleasure to be joined by Dr. Dieudonne Sankara from the World Health Organization. He and I will be co-chairing the session this morning. And hopefully you're in the right room, as we're going to talk about how we have a dragon by the tail and really talking about lessons learned from the eradication effort over more than 30 years and where we're going and what we're working on to try and reach the last mile in efforts to eradicate guinea worm. We're going to jump right into the session, and I would like to welcome Dr. Sarah Guagliardo. Dr. Guagliardo is an epidemiologist with the Centers for Disease Control, and she's assigned to the Carter Center, thankfully, to assist us in better understanding data, and she's going to take us through her session on managing and mining guinea worm surveillance data for action. You're very welcome.
Great. Good morning. Thank you, Adam, for the introduction, and thank all of you for getting up early and sticking around until the very last day of the conference. I'm excited to be here to talk to you a bit about our surveillance data and how we use it to inform our eradication program. My talk today is organized in 3 parts. First, I'll give you a bit of guinea worm background information. Then I will walk you through 2 examples of data analysis projects that we have, and I will leave you with some thoughts about challenges and possible solutions to those challenges. So, guinea worm has been around for a very long time. It was mentioned in Eber's Papyrus, which is an ancient Egyptian medical text dating back to 1550 BC. They've also identified calcified guinea worms in ancient Egyptian mummies. And guinea worm was also portrayed in a medieval painting of Saint Roche, who somewhat formidably is the patron saint of dogs. Since the mid-1980s, when the guinea worm eradication program began, we've seen a 99% reduction in cases, representing one of the great public health success stories of our time. And that success is largely attributable to improved access to safe drinking water around the world, but it's also attributable to efforts of many ministries of health supported by the Carter Center and WHO. So we've made a great deal of progress, but we're not quite there yet. This year, we have registered a total of 48 human cases in 4 countries. The majority of these cases have occurred in Chad, and as you can see from the table within Chad, the majority of the infections are occurring in dogs, which is the reason why most of our surveillance efforts today are focused on dog infections, and that's also why my talk today is focused on infections in dogs in Chad. Transmission begins when a person ingests contaminated water that contains infected copepods. Once inside the human host, the male and female larvae mate, and then over the course of the next 10 to 14 months, the pregnant female worms then mature, and they can grow up to a length of up to 3 feet, which is why one of the nicknames of guinea worm is little dragons, which is also part of our symposium title today. Eventually, the adult female worm will migrate to the cutaneous tissues, usually on a lower limb, but a worm can emerge at any part of the body and it can cause severe pain and disability. And I have some pictures of typical guinea worm emergence events on feet here. But eventually a blister forms, causing severe burning and itching, and that motivates the host to seek relief by submerging the lesion or the blister in water. And then upon contact with the water, the anterior end of the worm, she sort of pokes her head out and then releases up to 500,000 larvae into the water. And then those first stage larvae are then quickly gobbled up by hungry copepods, and the cycle starts anew. You can see a guinea worm larva inside a copepod there. In 2012, we discovered that dogs in Chad were being infected with guinea worm. There's still some degree of biological mystery about how this is happening, but we believe that fish are involved as possible transport hosts. To further investigate those transmission dynamics, we support a robust research agenda with many university partners, as well as with several groups within CDC. There are a number of interventions that are designed to interrupt transmission at various points on the transmission cycle. The few that I'll highlight here today involve case containment. What we mean when we talk about containment is we have effectively prevented a case or a dog from entering water and then contaminating a new water source. So for people, when we find a human case of guinea worm, we educate them about guinea worm prevention, and then we also encourage them to remain in a health center until their lesion has completely healed. The canine equivalent of that is that we tether dogs to a stationary object, usually a fence post or a tree, until the lesions on that dog have also fully healed to prevent it from entering water. The last thing I'll highlight here is that our reporting system is fueled by a cash reward program whereby individuals, and it could be anybody in the community, report possible cases of guinea worm to our program for further investigation. And if those cases are confirmed, those individuals will receive a monetary reward to incentivize reporting structures. Guinea worm surveillance in Chad is quite extensive. As of this week, we have 2,300 villages under active surveillance, covering a total of about 87,000 dogs. To my knowledge, this is the world's largest active surveillance system that simultaneously searches for cases of disease in both humans and animals, and live animals, I should mention. And I recognize that's somewhat of a specific category, but nevertheless, I think we have it covered. You'll notice that the majority of villages under active surveillance are located along the Shari River, which spans from Lake Chad in the northwest part of this map down to the southeastern border, and that simply follows the epidemiologic pattern that we see. And the remainder of the country is covered by a passive surveillance system. The Chad Guinea Worm Eradication Program is situated within a larger Ministry of Public Health structure, which is everything that you see in this blue box here. And our active case finding efforts really originate with the village volunteer. So we have at least 2 volunteers for each village. It's 50% men and 50% women. For some of the larger villages, we have more than 2 volunteers. And these volunteers are responsible for conducting daily household screenings to search for cases of disease in both humans and animals. As an example of what case finding looks like in dogs, there's a picture here of a volunteer sort of running his hands over the surface of the dog and palpating to look for signs of subcutaneous worms. When a village volunteer finds a potential case, they notify our paid Guinea Worm Eradication Program paid staff, who in turn conducts a case investigation, fills out case forms, and conducts worm extraction. The lower photo is of a volunteer submerging a dog's lesion in a controlled environment in this bucket of water to cue the worm to poke out her interior end so that he can then initiate the extraction process in the dog. Case forms and information about these cases are shared with the larger Ministry of Public Health at the regional and national levels, as well as with the Chad Guinea Worm Eradication Program headquarters in Geneva. And in turn, the data and case forms are also shared with the WHO and Carter Center. Worm specimens are also shipped to CDC for laboratory confirmation via morphology. And when morphology results are ambiguous, they're then sent for PCR. And then CDC relays those results back to Carter Center and WHO, who in turn relay the results back to the field level so that the reporting individuals can then receive their cash reward. So every time we identify a possible case of guinea worm in Chad, we collect a number of pieces of information, including information about hosts, worms, owners of animals, and villages. So examples of the type of information that we might collect about human hosts or owners of animals include demographic information like age, sex, ethnicity, and occupation. But for owners of animals, we also interview them about the possible history of infection in their dog. Examples of worm-level information might include the date that the worm was initially detected and reported, the date it was extracted, and whether or not that extraction was successful. So how can we use surveillance data? I like to think of the use and application of surveillance data as occurring in sort of this upward spiral of improvement, whereby you first collect the data, and then you put it to work, you analyze the data, and then once you've analyzed it and actually used it, you've come up with ideas about how to improve the system and to improve your data collection processes. But of course, when you're analyzing data, there's two main outputs that I can think of. The first being you take public health action. Of course, that's the most important. So you identify cases, and then you contain them. You make sure that water sources are not being contaminated, you ramp up your health education programs, and you mount a public health response. But you also might be able to identify new research questions and new ideas that come out of your initial data analysis that are perhaps better addressed in using other methodological approaches that are specifically set up to test hypotheses, such as case control studies or even experimental approaches. With that said, I have two data analysis examples that I'd like to walk you through. First is a descriptive analysis of infected dogs in Chad, and the second is a worm burden analysis of infected dogs. And then I'll briefly mention some future and ongoing projects. So I wanted to show you what the typical profile of an infected dog looks like in Chad. I think our lives would be a lot easier if all dogs in Chad had Facebook accounts because we could more easily track their information. In any case, a typical profile of an infected dog looks something like this. They're about 24 months old. They have an average of 1.8 worms per dog. A slight majority of these dogs are male, and the vast majority are used for guarding homes. So although relationships between people and dogs are different in Chad, with that said, dogs in Chad are valued, and every dog in our data set has an identified owner and a name. About 30% of dogs had a history of guinea worm-like infection in the same year, and 13% had a history of guinea worm-like infection in a different year. And of these findings, to me, these last 2 points are most interesting. In the first case, 30% of dogs having a history of infection within the same year, to me that indicates a possible issue with sensitivity. So it could be that we are detecting worms that or dogs that have multiple worm infections, and we're missing that first worm, and we're not getting them in the system until their second worm. And then in the second case, we have dogs that are experiencing recurrent infections. I think this raises a lot of questions about dog behavior and what those dogs are doing to continually experience infection. The profile of a typical dog owner. The average dog owner has 2.3 dogs, with about 35% of dog owners having just one dog. 26% of dog owners were of the Sarakaba ethnicity, which is a very common ethnicity in this region. And 75%, or close to 75%, were in farming-related occupations, with 17% being fishermen, and then a very small proportion of dog owners were in hunting-related occupations. I'll briefly mention that the reason why we're interested in occupation is, again, because of the possible exposures to fish and relationships with fishing. Spatially, we see two main foci of transmission in the northwest and southeast of the Shari River. And then in time, transmission is also very focal, so the vast majority of cases in dogs occur between the months of April through August, with a peak in June. And again, we believe that's related to fishing patterns, which in turn are driven by precipitation patterns and river levels and concentration of fish. So how can we put this descriptive data to work? So to me, the most interesting thing that we found is this theme of clustering that's occurring both in space and time and then within canine hosts. And that actually gives me quite a bit of hope, because I think it means that we can be smarter about targeting our interventions, ramping up surveillance in these key areas at certain times of year to utilize our resources more effectively. A number of research questions have also come up I think it's really important to dive into the underlying processes that are driving these broad-scale patterns. And I also think that there's a lot of room for an anthropological lens to better understand people's relationships with dogs and how they care for dogs. And then from the standpoint of improving surveillance processes, I think sensitivity should be an area of focus moving forward, and I'll talk a little bit about that towards the end. In a second analysis, we ran a series of negative binomial regression models predicting the number of worms that a dog might have. The reason why we might be interested in high worm burden infections is because these animals have the potential to contaminate many water sources over a longer period of time. The photo you see here is of a dog with multiple worms, and you can see that in the foreground a worm is actually being extracted, and there's several lesions from worms that were extracted in that back limb there. So most dogs have just one worm, but that range can go up to 79 worms. There was a poor dog in 2016 that presented with 79 worms, and unfortunately she passed away shortly after we detected her in the system. But factors increasing risk for worm burden included prior history of guinea worm. So I thought that was really interesting. So these animals that tend to have high worm burden infections are also likely to be infected in subsequent years, which again raises questions about the behavior of these dogs that are repeatedly being infected. And it also raises questions about the underlying host biology. Factors decreasing risk included owner occupation and the number of dogs in the household. I think these associations are a little bit more difficult to explain, but it's possible that it's related to the amount of infective inocula that dogs are receiving in different environments. The action points, I think, from this, the fact that we discovered that high worm burden dogs are also more likely to be infected over time really underscores to me the importance of monitoring these animals over time. From the surveillance perspective, we're now having conversations about the possibility of microchipping dogs, perhaps in certain high-risk areas, to really understand over time what their behavior and risk factors might be. The analyses I presented here are just the tip of the iceberg, but since we're talking about parasites, I think a more appropriate metaphor would be the head of the 3-foot-long worm. But in any case, we have a number of ongoing projects and future projects that are planned, the first of which is an analysis of recurrent infection in dogs. And as I mentioned before, the program does not track individual animals over time, so the only way we can identify these recurrent infections is by setting up a fuzzy matching algorithm in R to identify dog names and owner names that repeatedly show up over the course of several years. I can say that of the 1,931 unique dog names in this data set, the top 3 most common dog names in Chad are patients, police, and support. So we look forward to presenting the results from that perhaps next year. We also have a project in collaboration with CDC's Geospatial Research Analysis and Services Program, looking at risk mapping for guinea worm, and the question of interest there is, what are the areas within Chad that could ecologically support transmission, but where we do not have active surveillance set up? I'm particularly interested in this part of southern Chad, that to my eyes appears to be ecologically very similar to the Shari River area, but I think that'll be a very valuable exercise and will help us be a bit more proactive about finding cases. I think there's also a lot of room to combine the epidemiologic data with population genetics approaches. In a concrete example of that, I'll be teaming up with Dr. Liz Thiele, who you'll hear from momentarily, and Carmen Unterwegner to take a deeper look into the data that came out of an outbreak that occurred in Salamat region to determine whether or not that situation is unique and what we can learn from it. There are many challenges to surveillance. The 2 that I'll mention here are sensitivity and informatics. And as an illustration of some of the issues that we have for sensitivity, I would like to challenge you to a game of Find the Worm. You see several sets of dog paws here, some of which look a bit healthier than others. But if you consider your options carefully and you zeroed in on the picture in the upper left-hand corner, you were correct. If you look carefully, there's a forming nodule, and it's very subtle and difficult to see, and that's one of the challenges that our field teams face. In fact, I think a couple hours, even minutes after this photo was taken, a guinea worm did emerge on this dog. So to improve our sensitivity, we've conducted a number of field staff trainings. The photo you see in the middle there is of our veterinarian, Dr. Dieudonne, who is teaching a field supervisor how to appropriately palpate the dog to search for signs of worms. Dr. Jeff Priest at CDC is also in the process of developing a serologic test to detect pre-emergent worms in dogs. Sensitivity and specificity look pretty good for that test, but we're currently in conversations about how to best operationalize that in the field. And then lastly, we are now in discussions about initiating canine case sweeps in villages, and by that we mean we would gather a team of animal health experts and veterinarians to thoroughly search every dog in some villages a couple of times a year to supplement the volunteer-based system. I think the fundamental conflict that we're having there is that the volunteer system was set up to detect cases of disease in humans, and it works very well to do that. And now we're sort of pivoting and trying to focus on animals. We have a number of informatics challenges. Historically, we've used a paper-based system and entered data into Microsoft Excel spreadsheets that are not linked together in a necessarily helpful fashion. So we're in the process now of transitioning to an electronic data capture system using a software called Nemo, developed by Dottie Hunt at the Carter Center. And eventually the goal is to transition to a true relational database where data are stored in a cloud and can be accessed by multiple parties in real time. Lastly, we've recently engaged with the CDC Informatics Fellowship Program to conduct an independent assessment of our data flow processes in informatics, and that's been very helpful. So I'll leave you with some final thoughts. From an analytic perspective, the main takeaways are that we observed this clustering infection over space-time and within hosts, which leads us to think that we can better target our interventions. And I think we need to be more vigilant about monitoring high worm burden dogs. For the next year or so, our plans are to focus on improving sensitivity and informatics for our surveillance system. And with that, I will say that the work I presented here today is situated within a much larger community of public health practitioners. In a few moments, we'll be hearing from Dr. Makoy Samuel about some of the special challenges of running an eradication program in South Sudan. And Dr. Liz Thiele will talk about her work in population genetics, which I think are really fundamental to understanding population dynamics. And then lastly, we'll hear from Dr. Dieudonne Sankara about the special challenges of certification of eradication in the face of a zoonotic parasite. And with that, I will thank my colleagues, in particular Dr. Chindabet, who's our Ministry of Public Health guinea worm lead, the guinea worm team at the Carter Center, many of whom are here today, and Dr. Sharon Roy at CDC. So, thank you.
Thank you, Dr. Guagliardo, for your great presentation. I think the format of how we'd like to run things today, after each presenter, you can have any kind of hot topic questions that you might have. We'll entertain at least one question, and then if anyone else has questions, we will have time at the end of the session where you can ask anyone on the panel any other questions you might have.
So, hi, that was a great talk. This is Ruben Granch. I'm with Westergaard. And I had a question about time, because I see at the end that you're talking about using more real time. And often in the past, we collect data, we take it home, we hoard it for a little while, and then a year later we come out with reports and these sorts of things. So can you speak to that time element, and are you using mobile technology, or are you getting real time? And what's that turnaround time to the field teams in terms of the data they're collecting, and also for the compensation as well?
Yeah, that's a good question. So to clarify, we're still primarily paper-based. We've only recently deployed about 300 phones to do some pilot work for data collection. So right now, we're not using electronic data, but I envision within the next couple of months we will get there. With the paper-based system, right now, the turnaround time can be slower than we'd like, but part of that is also just the nature of guinea worms. So we identify a possible case, most commonly in dogs, and that information, preliminary information, is reported up to the national office in Geneva, and then there's this follow-up period because the dog needs to be observed, right? So it can be in some situations weeks before we have a final pieces of information about that case. And then our data, finalized data, take a total of 30 days to verify, if that makes sense. And we're hoping that when we transition to electronic data, that might help reduce redundancies and the need to call people in the field and verify that sort of information.
Great. One thing to add is we're going to attach something we call SmartNet to our malaria nets. And it's a short code and allows 2G mobile technology, and we're hoping to crowdsource information directly from the community. And I mean, you probably already thought these things through, but that would be something as well, where if you're offering compensation to people for finding cases, then using short codes and mobile technology and these sorts of things can allow you for much quicker response time. I don't know how to shorten the other times in terms of verifying worms and these sorts of things, but definitely technology is getting to the point where we can disintermediate a lot of the other more clunky methods of collecting data. But keep up the great work. It's great.
Well, thank you. And thanks for your comment.
And we'll take one more quick question, and then we'll move to the next presenter. Thanks.
Hi, Dominic Lucero with the Clinton Health Access Initiative.
Hi.
I noticed on one of the maps, the foci of cases go right pretty much onto the border of the CAR. And so I was wondering if there's any surveillance work to try and scope out how far it might go. I know it's maybe not safe to send folks into CAR right now, but just sort of wondering what sort of surveillance activities are going on around that.
That's a very good question and an astute observation. I think we're all concerned about those cases that are occurring near the border of CAR. Right now, we don't have any surveillance activity in CAR because, as you mentioned, there's significant security challenges there. However, that is one of the purposes of going through our risk mapping exercise. We're starting that activity with Chad, but eventually we may consider expanding it to a larger region to see if we can identify pockets of risk outside of Chad and areas near Chad. But I think we're all aware of that concern, and it is on our radar. But yeah, it's a good point. Yep. Great. Thank you.
Thank you so much. Thank you, Sarah. We'd like to welcome Mr. Makoy Samuel Yibi to present on the art of multidisciplinary actions interrupting transmission among highly mobile population in conflict and post-conflict areas. Mr. Makoy is the director of Guinea Eradication Program in South Sudan, and also the director of for preventive chemotherapy, neglected tropical diseases within the South Sudan Ministry of Health. Makoy, the floor is yours.
Thank you, Dr. Sankara, and good morning, everyone. The dragon in South Sudan, in our view, is going down. And it's not a question of if, it is a question of when. And for us to be able to appreciate the achievements in the South Sudan Guinea Eradication Program, we need to be able to probably work together and work back to be able to see the challenges on the road from the start of the eradication campaign. And I want to mention those challenges in terms of a big picture. I think all those who have been monitoring the eradication campaign in South Sudan, you realize things like insecurity is a big picture. And definitely for obvious reasons, people at the time are convinced that the insecurity has become a huge impediment for the eradication campaign in Sudan way back to 1993. That's when the campaign started, when South Sudan was still part of Sudan. And at the time, there were only a few locations that were accessible when the National Active Case Search was initiated in Sudan. And already at the time, there were nearly 3,000 cases detected. That was 1993. And clearly, there was no way to think in terms of establishing a structure to be able to do surveillance and to maintain that structure across every part of South Sudan, because as we know, by the time there was the civil war, and most of the parts of the South Sudan at the time was considered liberated areas. So the areas that were accessed by the government of Sudan at the time were only what were considered the garrison towns of Juba, Malakal, and Wau, and the immediate surrounding. So clearly, it was viewed as a clear, major hurdle to the global eradication campaign. But then the other thing that I think at the time, when you mention South Sudan in terms of in relation to the Guinea eradication program, and then you start to think in terms of the burden of the disease. And by the end of 1994, already South Sudan, in fact Sudan, reported more cases than any of the 19 endemic countries at the time. But I think it is important to mention that the majority of those cases were actually from South Sudan. If there are a few cases detected in the northern states, these are probably importation also from the south to the north. So clearly the issue of burden of disease was one thing that stands out. And I think the other thing that is important to mention in the journey of the eradication campaign in South Sudan is the Guinea ceasefire. I think it was clear that had it not the Guinea ceasefire, probably the narrative for the Guinea campaign in South Sudan would have been different. It enabled a situation where at least it was viewed that something could be done. Some good action, good actions could be able to at least generate something. We know that at the time the negotiations were quite intense. And I think even people at the warring parties are very committed to finish each other. And of course, take down the population with them. And I think it was not only the issue, I think they could say, okay, let's agree on ceasefire, but they were actually not happy to decide what time to effect the ceasefire. Because the rebels at the time, who are now the ruling party in the country, wanted the ceasefire to be in the rainy season, because they don't want to be fighting in the dry season, because the government will employ all the machinery they have. And the government in Sudan wanted the ceasefire to be not in the dry season, because they wanted to wage war in the dry season, which they will have an advantage. So it was quite an intense negotiation. Well, President Carter did it, and it came to effect. I think one of the correspondents in Egypt had made this notion that usually the dove is a sign of peace. But in Sudan, at the time, it is the worm that gets attention. So this Guinea ceasefire definitely created a scenario where we know that after 2005, with all these hit and run interventions between 1995 and the time that we have the peace agreement, already cases definitely came down. We know that in 1996 we had over 100,000 cases immediately because of the opportunity to do the countrywide active case search in 1995. So the peace agreement definitely provided an opportunity for the country to be able to establish the much needed surveillance structures and to be able to make sure that we're able to have a clear agenda of interrupting transmission. Well, with the ceasefire came the opportunity now to establish the Guinea eradication program for the southern part of the country, for the southern states. When it was established, it was called the Southern Sudan Guinea Worm Eradication Program, and now it is the South Sudan Guinea Eradication Program, especially after the vote for independence in 2011. It is important to note that when we were establishing this program, there were no structures, there were no institutions, there was nothing to relate to. So basically, whatever structures were established from the scratch. And I think it was probably a blessing in disguise, because at that point, the structure did not belong to the Ministry of Health. The genome key structure for eradication belongs to the community, and the community became the key stakeholder in the fight against guinea worm. Now, we realized at the end of 2006, we had reported over 20,000 cases, and we continued to see a reduction in cases, and we maintained that reduction of cases until 2011, and with the independence came also a renewed energy, and with that renewed energy, we were able to surge forward, and in 2013, for the first time, we were able to report only a 3-digit number of cases. But then we started to see a very strange scenario. We were starting to see cases appearing in counties where we thought we have interrupted transmission. And then we thought, okay, maybe these are just isolated situations. Maybe our surveillance missed to pick up something. Okay, let's respond to those areas, and we sort of quite effective in normally going back and clear those pockets quickly. And then we continue to see the same situations of having quite weird, strange cases appearing in areas where you cannot actually link it to where we expect to see cases. And then in 2015, something completely new happened in the context of South Sudan. For the first time, we reported a dog infection here. Only one dog infection in the same family where we have one human case. Nothing else. No any other dogs. We actually went to do a dog census. We counted over thousands of dogs and tried to really intensively check on dogs as we do our daily surveillance, but that's it. And 2016, we basically finished the remaining foci, and in 2017, we were all celebrating when over 15 months, 0 cases, and we thought that, okay, it's done. Well, in 2018, we will also witness some surprises. Well, what is so surprising here is that when you see the different circles where we have cases in 2018, there is no epilink. It's like every location is an island. You can actually investigate everybody, and it is difficult for you to be able to get an indication that there were missed cases in the previous year. At least in this area, in the area of Lake State, because it was quite an insecure area, so for some reason we felt that maybe we missed something because of issue of insecurity. Maybe we missed something because we are dealing with mobile population. In here and here is quite a mystery. Well, see what happened in 2019. All the 1+ villages that accounted for cases in 2018 reported 0 in 2019. And then we see 2 foci, one here in Udishi, Western Bahar Ghazal, and one here in Torit. The Torit one is a mystery, because in that village, you cannot even remember, since we started the campaign, you cannot even remember when was the last case. You can ask anybody, including the others, like the elderly people, and they will not even remember when was the last case they saw somebody with guinea worm. So we think that definitely there seems to be a low-level transmission outside humans, where humans become accidental hosts. So we think that from our investigation, we are actually seeing a situation like here, especially in this situation of Udishi here in Barurud, in Akwayo. It's very typical here is that the pattern of settlement is such that households are actually having more interaction probably with the animals than with the other humans, because the water sources they use are actually more specific to individual households in the middle of a forest. And really, you don't know the other users of those water sources. So, but all in all, we believe that there is a pattern of who are the at-risk groups right this time around in South Sudan. is always the population who are mobile, young people, women who are mainly the breadwinner and involved in fetching water, villages that are very, very remote. Sometimes those households where we see cases are actually sort of like really remote compared to the other households in the same village. So we are actually seeing a pattern here, a pattern where you are seeing cases, isolated cases happening in households that are quite at the periphery. Probably there is more interaction, more with some wild animals, especially as they compete in, on the crops and also water sources that are used by these households. But of course, this age group particular brings in a number of other dynamics. Access becomes an issue. Insecurity is an issue, especially when you have young people with guns. And definitely, that's where we believe that even if you can be able to provide safe water at the household level, this population is going to go away from where tens of miles away from the households to be able to find grazing and water for the animals. So as a program, what we did was try to think of solutions. One, over a few years, we have started really to think critically of how we can be able to build a flexible surveillance structure, a structure that is embedded on the mobile population. We recruit, we train, and we embed this team with the mobile population. Where the population move, this team move with them, they continue with awareness, they continue with detection of cases, they continue with the interventions. We have also continued to engage with local leaders and also political leaders to be able to help create an enabling environment, especially when we hear that there is a disarmament of these young men with the guns, and then we always try to work with the leaders so that we are just behind the line of those doing the disarmament, and when the opportunity avails itself, we move in and we access those populations and with whatever we got to be able to understand if there is any missed cases, and also to be able to provide the necessary awareness. So with this, we believe there are lessons to learn. We think that even in this insecure setting, interruption of transmission of guinea disease is possible. I think when we started in 2006, we know that it was going to be daunting, but it took commitment towards commitment and resilience for us to be able to conquer the challenges, geographical, logistical, and other challenges. And I think at the end of the day, making sure that the community, we built a relationship which is actually built around all endemic villages. In fact, we built a relationship where the program and the community is like a fish and water. Our survival of the program depends on the community. And I think that is very, very critical for us to be able to relate to the different challenges in different locations. I think we also felt that the strong leadership was very, very critical. I think we are so lucky because, well, when I talk about guinea worm, the president of the republic understands what guinea worm is. Just 10 years ago, he was a commander in the field. He knows what guinea worm is. He has seen his soldiers suffering with guinea worm. So it was not difficult for us to be able to mobilize that attention from the leaders. So that leadership is very, very important, and I think it also put the program at its toes because of the expectation from the leadership for us to be able to do what is required. And I think in the same way, we also, as a program, we try to make sure that we address the issue of technical leadership for the program. And we definitely built a partnership around everything that we do as a program, and I think that partnership is very critical. We have the secretariat, which is really the decision-making body of the program. We meet every week to be able to devise technical and operational mechanisms that could enhance the work in the field. We ensure that every challenge should be an opportunity for us to learn and to learn lessons, and also to be able to find solutions to address whatever challenge we faced in any given time. We also, in 2018, program is now being strengthened with the establishment of the South Sudan National Committee for Documentation, Dracunculus Elimination, which is basically the elimination committee for South Sudan. And we think that the investment in building up this structure over the last 10 years has really provided an opportunity for the country to build up a system. We have a clear statistic that shows that the current formal system only serves less than 50% of the population. So the Guinea structure was viewed by the leadership of the ministry that this is the structure for the majority of the people who don't have access to health services. And I think this is looking at the structure we have at the village level, the village volunteers, the area supervisors, the field officers, the program officers, and the technical advisors who had actually done a good job training the program officers. And now we have a situation where program officers are actually now taking the role of technical advisors. And I think that has really enabled the Minister of Health to appreciate the fact that there is already a system that the ministry can leverage to be able to ensure that universal health coverage are addressed. The Burma Health Initiative as a result was formed from that perspective, and based on this, communities like this that have no health services are able to get a package of these basic health care services. And in summary, I wanted to take you through this journey where we had huge burden of the disease, mounting challenges to be able to bring it down. We think that with the current isolated challenges, we still are solid and pursuing the direction to ensure that in a very short time, we are able to interrupt transmission in South Sudan. We were, just before we came here, we were in a process to make a presentation to the Council of Ministers shared by the President of the Republic. And we had some few things to present to the leadership to address. The issue of boreholes. We think that still it is important that these communities have access to boreholes, but more important, we are actually trying to ask the leaders to help us improve access to areas affected by conflict. There is also the Fulani nomads who for the first time we were able to reach them this year through a collaborative action where we're doing active case searches with mass drug administration, and the team are currently in Raja doing the same exercise. And we also wanted also to access them borrow and try to assess them. It's important to note that these people traverse all the border through Chad to West Africa. They are in the bush. They don't come to towns. When they come to towns, they only come to sell a few things. So we think that probably we need to be able to understand what is happening with this group. So with this, I want to thank everyone for your attention, and I want to acknowledge the support from the Carter Center, the World Health Organization, UNICEF, the Ministry of Water Resources and Irrigation, the Guinea warriors, some of them are here, and of course those who are supporting all our effort. Thank you very much.
Thank you very much, Makoy, indeed. One burning question. If not, we'll pull all the questions at the end for the matter of saving time. Okay, good. So I would like to welcome Dr. Thiele to present on covering all the bases using population genetics to elucidate medinensis, transmission, dynamics, and population history. Dr. Elizabeth Thiele is a visiting scholar in the biology department at Vassar College and serves as a technical consultant to the Carter Center's Geneva Eradication program. And Dr. Thiele generates, manages, and analyzes genetic data on Guinea worm populations with a particular focus on understanding transmission dynamics. Welcome, Dr. Thiele.
Thank you, Dieudonne. Good morning. Today I'm going to tell you about our efforts to support Guinea worm eradication program using genetic tools and population genetic and evolutionary theory. We'll cover some specific results, but also take kind of a 30,000-foot view of how we're working to incorporate the genetic data and discuss some of the challenges that the Guinea worm system poses to genetic analysis. In 2010, after a period of almost 10 years of zero case reporting, guinea worm resurfaced in Chad, and in 2012, the first emerging worms in dogs were officially detected and collected. The first and most obvious question was whether that worms emerging from dogs were truly guinea worm or a related dracunculid nematode similar to the North American species Dracunculus insignis, which is known to occasionally infect dogs. These worms were submitted to the CDC and in Atlanta for morphological and molecular evaluation and sequencing of the highly conserved 18S small subunit ribosomal and cytochrome C oxidase genes, as well as whole genome sequencing by collaborators at the Wellcome Sanger Institute in the UK, indicated that the worms collected from dogs were in fact Dracunculus medinensis. So upon determining that the worms collected from dogs were guinea worms, and the eradication program's continued detection of infections in both dogs and humans, the natural next questions were, first, what caused this apparent reemergence of guinea worm in Chad? Was there a reintroduction from one or other endemic countries, or recrudescence from one or several refugia that had escaped notice previously? And although worms were collected from dogs, and they were determined to be Dracunculus medinensis, were they from the same population of worms infecting humans, or were dogs perhaps maintaining a separate population of worms, isolated from human population by biologically or epidemiologically unique factors? So perhaps were they maintaining a cryptic species that was indistinguishable from human worms by morphology and highly conserved sequences, but effectively maintaining a separate cycle of transmission from the human worms. So to answer those questions, we turned to population genetics and the associated molecular tools. Specifically, we developed and implemented two molecular protocols. First, detecting mitochondrial sequence variation at 4 genes for population studies, the cytochrome B, cytochrome C oxidase subunit 3, and NADH dehydrogenase 3 and 5 genes, representing approximately 20% of the mitochondrial genome. Cytochrome C oxidase subunit 1 was also included for interspecific phylogenetic inquiries. And the rationale for using mitochondrial sequence variation was that, one, as maternally inherited marker, it would allow us to track maternal lineages, and as the emerging uncontained female worm is the agent of disease transmission, would therefore allow us to generate kind of a conservative estimate of uncontained infections. In other words, infections that were not being reported to or detected by program staff prior to worm emergence or exposure to water bodies, if at all. Additionally, mitochondrial sequence variation is a useful population genetic tool for detecting differences between populations over large spatial and temporal scales, and is also commonly used for detecting cryptic speciation. It's conserved enough to be species specific, but is more mutable than the conserved nuclear genes. We also used microsatellite length polymorphism at 23 tri- and tetranucleotide loci, which, thanks to their highly variable nature, are much better at differentiating between individuals at smaller scales, both spatially and temporally. This allows for inference of local population dynamics, such as recent population history and demography, and the estimation of kinship between individuals. In addition, because microsatellites as nuclear markers are inherited from both parents, as demonstrate here, both mom and dad, they allow a window into the genetic variation that exists in adult male worms, which are never physically sampled in natural populations. So, obviously, one of the most burning questions about the canine fiery serpent has been, are dog worms and human worms the same? And do they share the same parasite population? And the answer appears to be yes. The results of the work were published just over a little over a year ago, but we'll just kind of walk through real quickly. The overview and takeaway message is thus: the phylogenetic analysis of African guinea worm samples relative to their American dracunculid congeners using the cytochrome oxidase subunit 1 shows that all African guinea worms, regardless of host species from which they were collected, form a single strongly supported clade. Likewise, analysis of microsatellite variation within Chad finds that geographic origin of a worm is a stronger predictor of and explains significantly more genetic variants than host species. So here we're viewing a spatial principal correspondence or principal component analysis And we see that worms from the northwestern region here, represented here, are more genetically similar. So worms from a dog collected here and worms from humans collected here are more genetically similar to each other than, say, worms collected from a human here and a worm collected from a human in the southern region. And this pattern holds regardless of the analytical approach that we take to investigate parasite population structure. So where did these worms come from? Were they the result of a reintroduction or from one of the currently endemic countries? And again, the answer here, in this case, appears to be no. These appear to be Chadian worms. Not only is there no evidence that the Chadian worms were introduced from another contemporaneous population of guinea worm, but the Chadian guinea worm population appears to have been relatively stable through the years of zero case reporting. Both nuclear and mitochondrial data show that the Chad worms have considerably more genetic diversity than those from other endemic regions, which is kind of summarized here by this phylogenetic tree on the right. You see in red the diversity and extensive divergence in the Chadian populations relative to, say, samples collected from Mali or samples collected from Ethiopia or South Sudan. And furthermore, coalescent model-based demographic inferences illustrated by the Bayesian skyline plot here do suggest that while we do kind of see some evidence of decline in the population, it also appears to exclude the possibility that the population of worms either disappeared or were reduced by to a very small bottleneck during the decade without reported human cases. And I'll also note here that while the y-axis illustrates effective population size, these inferences were made with the mutation rate for C. elegans, as there's no direct measurement of mutation rate for guinea worm or any other related parasitic nematode. So while mutation rates are relatively consistent across eukaryotes with similar genome sizes, variation of several fold from the value we've assumed would not be very surprising. So these analyses should be interpreted as relative rather than absolute estimates. Now, in the absence of Chad samples prior to 2000 or more extensive sampling from neighboring countries, we also cannot exclude the possibility that the Chad worms we analyzed, which all emerged in Chad following the 10-year gap in reported cases, migrated from elsewhere or were imported from elsewhere. However, we see few Chad worms that are closely related to worms from any of the neighboring countries for which we had access to samples. And so this possibility is purely speculation and would seem that a quite large scale influx would be required to explain the level of diversity that we see in Chad by migration. So, questions answered, right? Case closed, I can move on. Just kidding. We're now undertaking a massively scaled up sampling and investigation of available guinea worm population genetics to try and understand more about the guinea worm's story. Quite literally, if it emerges and we can get it, retest it. Unfortunately, as far as we can tell, historical samples prior to 2000 are effectively impossible to come by, though if you know of any well-preserved specimens gathering dust on someone's shelf or in someone's drawer in their lab, please, please, please let us know. But while we have little historical genetic context, the goal is to generate as much genetic data as possible for as many samples as possible. The idea being that these genetic data can be interrogated and applied in various ways, particularly to help us understand more about what's going on in the guinea worm populations. For example, how might transmission contexts be unique among or even within endemic countries, and to supplement and augment classic epidemiological investigations and program monitoring that's being done by folks like Sarah and the field program staff. And so, for example, one of the analyses that we look at for patterns and dynamics is we see that Chad kind of follows a classic population paradigm in which relatedness between individuals generally decreases or decays over increasing distance. And so this would suggest that most transmission is relatively local in nature, which also kind of, again, corresponds with some of the observations that we've made just in terms of prevalence and the localized nature of prevalence. But on the other hand, we do have some initial analyses that indicate that some of the population dynamics may be regionally variable. So in the northwestern stretch of the Chari, relative to the southeastern portion of the population, we see much more admixture in the population and much more population structuring here in the southeastern corner. So one of the obvious questions is, do these patterns persist under additional investigation when we continue to add samples? And if so, can we identify factors that may be contributing to the patterns? So ecological or hydrological, because we've got more water bodies in the southeastern versus Chari River being the primary water body in the northwestern region, or are there cultural, economic, or even socio-political covariants that could explain some of these patterns? So additionally, in our investigation of one particular hotspot for dog infections, we've observed that even at the level of a single village, we continue to see high diversity of guinea worms in Chad. So here we've got a tree of mitochondrial haplotypes. And as you can see, there is considerable divergence between the haplotypes, which again corroborates the initial conclusion of a relatively large stable population, even in the time of zero case reporting. But note that even though we see high diversity, we also see just a few, in this case 2 or 3 of the haplotypes that are kind of overrepresented in the population. And as we continue to monitor guinea worm populations, we'll be watching for and would expect these less common or minor haplotypes to kind of drop out first and perhaps be an indicator of success. But I would note here that out of the 117 worms tested, the largest full SIB cluster that we detected was 6 worms. And on average, 3.5 dogs were infected by larvae from a full SIB cluster. So this means that on average in this village, the larvae from one uncontained worm went on to infect 3.5 worms. And you might say, but Liz, the majority of the worms shared the one of 2 haplotypes. So what does this mean? And for me, I think at this point it means that shared mitochondrial haplotype does not necessarily signify a direct transmission or epidemiological connection. It indicates shared maternal history, perhaps. Also, perhaps an indicator of homoplasy, and we're going to be working on expanding mitochondrial sequence sampling to kind of try and eliminate that possibility. And that such epidemiological linkages and making linkages between transmission cases will continue to require nuclear data and estimation of kinship. And obviously we're still collecting data both within this village and in other villages to see if these hold up for further investigation. But also of particular interest is to be able to feed these data into behavioral analyses or behavioral data sets that are being constructed by studies to monitor animal movement and water contact patterns. And so I'm hopeful that comparisons of parasite community and population genetics will, with host behavioral patterns, will maybe provide some additional insights into population dynamics in the non-human population. One of the biggest limitations of our guinea worm genetic data set, as I previously mentioned, is the lack of historical material. So moving forward, one of the more useful outcomes of our effort will be to create what is effectively a reference or catalog of guinea worm genetic variants, and this will be key to addressing the unexpected cases or cases of special concern that pop up and trying to place them into the context of the entire guinea worm population. So specifically, for example, much has been made of recent Angolan cases, and when we do phylogenetic analyses with mitochondrial data set for those, we see that of the 3 worms that were detected, there are 2 haplotypes, and they are placed in a highly supported cluster nested within a small, highly supported cluster of Chadian worms. To date, these Chadian haplotypes that cluster with the Angolan worms have been found in 7 parasites collected from dogs, cats, and humans in 2014 and 2016, and from multiple geographical regions within Chad. So there's no direct connection to Chad. And at this point, while the phylogenetic analysis would place it in a cluster with Chad, that shouldn't be interpreted as or implicated as a conclusion of importation from Chad, particularly in the absence of historical molecular samples. The phylogenetic analysis illustrates the likelihood that the Angolan lineages and the Chadian lineages share a recent common ancestor, but not whether that ancestor was located in Chad or Angola or elsewhere. Population genetic methodologies are also being employed to investigate outbreaks in endemic countries. So for example, in Ethiopia in 2017, there was an outbreak among farm workers in one area, and epidemiological investigations suggested that there was a single source of contamination or infection, and genetic investigations corroborated that. So with the plot here circled and highlighted in red, these are the cases involved in the outbreak, and they all were highly probable full SIBs in the analysis. Unfortunately, perhaps, this analysis also exonerated the suspected source of infection, which in this case was a dog, and unfortunately we haven't been able to identify the source of infection so far, meaning that it was either undetected or unsampled. And similar analyses, as Sarah mentioned, are currently underway to investigate suspected outbreak in Salamat, and currently pointing in the direction of a true point source outbreak, but we still need to do the nuclear genetic evaluation to make sure of that. Now, I would like to point out that guinea worm as a system, just in all of its aspects, presents some challenges, not only to the programs, but to genetic research. So here I'm going to plug some work for our colleagues at Sanger Institute. And they've done population genomics analyses with these samples, and the whole genome work corroborates our data, but this data also came at a cost in time, effort, and money because of the challenges of the system, which in this particular case are that it is very hard to get clean data out of the worms with a non-specific target or molecular tool, because you end up hitting everything but the worm. And so you end up hitting host DNA, you end up hitting contaminating microbes because of the way that the worm is collected and spends time hanging outside of the host. So that is a challenge that we face. Secondly, there's variable presence of larvae. Our colleagues at Sanger have also indicated that you can get very clean data from individual larvae that have been preserved on FTA cards. However, that variable presence of larvae due to the extraction protocol means that at this point our estimate is less than half of the samples actually have sufficient numbers of larvae to include. So that limits our sample size. But in addition, the actual presence of larvae then further muddies the water, because you're dealing with a pool of samples, a pool of individuals with distinct genotypes versus one single individual. And some initial work that was done by Dr. Cotton and his team shows also that we're likely, in some cases at least, we have no sense yet of proportion of cases, we're also likely to encounter cases of polyandry, so that you have potentially 3 genomes in the mix, 2 fathers, 1 mother, and then how that all gets assorted into the offspring. The final kind of challenge that I'll mention is the fact, and this is perhaps one of the most frustrating challenges both for our genetic assessment of impacts as well as for the epidemiological case tracking, is that there's a very long generation time of the parasite. It's a fundamental biological constraint, and that not only affects how long it takes to collect information on the program impacts, but also how influences how quickly the population will respond to program measures. This implies that it will take at least 1 year to observe any impact or even a 100% effective intervention, and in realistic cases, we would expect it to take several, possibly many years to see statistically significant changes in the genetics of the population. That said, this is not all doom and gloom. We go forward with clear eyes and full hearts, and we won't lose, and lots of pipette tips. In particular, we are in the process of adapting our protocol to a next-gen sequencing of the whole mitochondrial genome to both improve throughput and amount of per individual data content. And the hope is that will facilitate addition of data into other areas. And I would like to mention that despite the unique limitations and challenges, there is also a unique value in collecting the genetic data of a metazoan pathogen as it undergoes extirpation, elimination, and ultimately, we hope, eradication. We find ourselves in a position of having to race to catch up to the worm in order to understand many fundamentals of its basic biology that we thought we had understood, or at least understood well enough to get us across the finish line. And it's a hard but valuable lesson, and one that we hope other programs are heeding. And while I wouldn't necessarily argue that it's incumbent upon the programs themselves or their donors to fund the basic biological research to cover all the bases, I would propose that it is incumbent upon them to extol the value of such work and promote it. You never know when you will need it. And finally, much can be made of the value of the genetic data to support epidemiological interventions and program monitoring, but it's also very much the case that epidemiological data, along with other on-the-ground data, is of great value and support to the genetic data, especially regarding identifying meaningful patterns and influential factors, and in informing its utility for certification purposes. So while genetic data is certainly a powerful tool, it may well end up being a very important nail in the coffin of guinea worm, it is very unlikely to be the hammer. So with that, thank you very much. Acknowledge a bunch of people, most specifically the field teams that make all of this possible. Thank you.
Thank you, Dr. Thiele. So we wait for a question at the end, as already agreed. Thank you, Dr. Thiele, again. Thanks.
I would now like to introduce, again, Dr. Dieudonne Sankara, who's the team leader of the guinea worm eradication program within the World Health Organization based in Geneva. Dr. Sankara is an old hand in eradication efforts, having been the former national coordinator for Burkina Faso's guinea worm eradication program. He'll be walking us through the certification process that's required and some of the lessons learned so far and challenges that lie therein. You're most welcome, Dr. Sankara.
Thank you, Adam, and thank you everybody for being here so early in the morning to listen to guinea worm, how we do that. Let's say, as we all know, this is let's say, one of the very few diseases that have been set for eradication globally. First being smallpox, done, very good. Now we have guinea worm, polio, and also yaws on the pipe. Hope we get there. So this is the worm, and you have already seen this worm, let's say, with Sarah and Makoy, and also Elise. So I'm not going to stay here longer. You are also seeing the cycle of transmission updated from Sarah, on which we based to elaborate the strategy for eradication. And then let's move and save some few minutes, very precious. So I'm going to walk you through the criteria and also through history and see where we are, and we know where we are, but just a reminder, and then we see the challenges, how we deal with that. So eradication is achieved when endemic countries produce evidence of interruption of transmission, and optimal surveillance is in place to detect any occurrence of diseases, should it happen. And all countries get certified as free of guinea worm disease. This is how simply in 3 bullets, but not easy to achieve. 3 principal mechanisms was established to facilitate certification efforts. Let's say we have an international commission for the certification of Dracunculus eradication, ICCDE, charged with the evaluation of the evidence presented by countries claiming to be Dracunculus free and seeking WHO certification of eradication. I'm reading it fully because this is the basics. Sorry for reading my slide, but this is how we do it for these few slides. Make sure we all are on the same page. And most of you actually know already. A certification cell within the WHO is served as the secretary of ICCDE, and the composition of a worldwide panel expert from which international certification teams called ICT consultants or advisor are selected. The aim of ICTs is actually to visit countries that have requested certification from WHO, and then to assess the evidence demonstrates that indeed transmission has been interrupted and the surveillance is good. So, but to do that, you need a standard criteria for certification, because this is very essential to ensure international credibility for a claim that if say that this is eradicated, this is eliminated in a country, then it's really true. To have a formal mechanism for judging the success of recent national tuberculosis eradication programs, and to have a standard, effective procedure to identify and eradicate any previously unknown foci of transmission, like in Angola, and to help investigate remote or sporadic occurrence of the infection in unconfirmed, potentially endemic areas. So, criteria for certification is what? You have to have evidence, again, of absence of transmission ensured by a minimum of 3 years of pre-certification stage, and evidence of functional surveillance system to prevent re-establishment of local transmission from any imported case, verified by independent external evaluation, visit of international certification team, and review by ICCD, which recommend or not recommend to WHO Director General to certify the country. This is actually the same like we did for the smallpox. Let's go through quickly the 3 years of pre-certification phase, which is crucial, as we know. The country, after interrupted transmission, should maintain the sensitivity of case detection by sustaining high degree of public awareness of tuberculosis, and stressing the importance of reporting cases, establishing a reward system, responding quickly to any declaration of suspect case, conduct awareness campaign and assess regularly the awareness penetration in the community, and integrate surveillance with any field presence, any program in the field, like IDSR, vaccination, MDAs for anything. Compulsory notification of cases and rumors by all units, health units in the country. So, and a rumor register should be maintained, and also at least one active case should be conducted nationwide, village by village. And surveys conducted on schools, market, everywhere. This is very intensive to ensure that there is nothing left unturned in the country before you go for certification. This is a summary of what I just said, to put a visual on what I said. And you have then, where am I? Doesn't work? Okay. Intervention in progress right now in Chad and South Sudan. And then transmission interrupted. Again, I'm repeating to make sure it is clear. Transmission interrupted, but now look, we have some animals, they are cautioned. Because so they have to take into account all these animals. Transmission interrupted, that means in any host, is what it means. Not only in humans, because as Liz said, it's the same parasite. And once this is interrupted, you need to go through 3 years of minimum of 3 years for surveillance. And then you get certified. But after the last country is certified, we need to wait a minimum of 3 years before declaring global certification of a disease. This will, the road is very sinuous, but we're getting there. We're close, actually. So, let's give you some background historical data on how we started, not how we started, but where we are, how we started and where we are. In the 1980s, after the smallpox eradication, the global public health leaders, including CDC and WHO, and started look, and others, I cannot say everybody, and some of people are here. Frank Richard know more history than me. He's sitting here. CDC at this time, they decided to go. Let's finish the Guinea worm disease. It's possible. And then, actually, let's move quickly. They took advantage of the decade of safe water, decade 1981 to 1990s, to have a first WHO World Health Organization, World Health Assembly resolution on Guinea worm. Oh, we can do that. Let's go ahead. Guinea worm is an indicator of safe water provision, which is good. And then, let's go. In 1986, Carrier Center created, and since then, South country is at the forefront of Guinea worm eradication. But listen, even people at Carrier Center who's doing Guinea worm disease, they also were actually at the CDC as well. So the same people are at CDC. Not that Carrier Center started on 1986 and started doing. People at Carrier Center at this time actually were also at CDC, who initiated the thing with WHO and other partners. So it's a continuous. Then we have many resolutions from WHO, whatever assembly, to support this. You need this one. If you don't have resolution from WHO, you don't get anything done at the global level. It's very important to recall this one. Let me go for sake of time. And in 1991, let's say 1986, sorry, we have another resolution at WHO asking for elimination of dracunculus. 1991. So this is the first time WHO requests for eradication of dracunculus. And then, following that, let's say following that, but let's put it, let's get it straightforward. From 1983 to 1993, all countries set up national programs. All, South Sudan came later on. But apart from Angola, which doesn't know they have Guinea, they were also in a war situation. So let's put it. And then our first certification of a country, let's say 1995, the ICCDE was created. And Pakistan stopped transmission in 1993. Kenya followed in 1994, actually zero case in 1995. India, 1996, stopped the transmission. Cameroon and Senegal and Yemen followed 1998. CAR, 2001. Uganda, 2003. Benin, Mauritania, 2004. Burkina Faso, 2006, including Togo also, and Cote d'Ivoire. Nigeria, 2008. Nigeria, we have one warrior here. William worked in Nigeria in 1774. 1976 onward. Thank you for your hard work there. Contribute to soft transmission. And Ghana, 2010. Listen, Nigeria has like more than about 700,000 cases a year. Ghana, more than, I don't even remember, but more than 200,000 cases a year. And this time, to tell you the extent of the problems. Sudan, 2013. And then no country has stopped transmission since then. This is where we are right now, with the red being the country currently endemic for the diseases. But if you see very well, apart from South Sudan, where there is no evidence of animal infection, you have Ethiopia, Chad, and Mali. They have all evidence of animal infection. It's not by hazard they're the last country being there and hanging there and giving us hard time to finish the job. And Angola also had some evidence of animal infection. But we have DRC. I see Professor Antoinette, who is sitting in the room, which is the chair of national certification team in DRC. DRC have no evidence of any infection. Then they're still working to complete the dossier for certification. So we have only 7 countries remaining to be certified, as I just listed here. And with 4 endemic countries, Angola is still yet to be declared endemic. And then with Sudan, finished, northern Sudan, but Sudan finished, but still need to complete the dossier. This is where we are right now. Very serious road, but a lot of things has been achieved. Since the global program, all countries need to be certified. And in another word, we have now 199 countries, areas, and territories certified, including 187 countries, which include the 16 previously endemic countries. This is where we are in the world in terms of certification of Guinea worm. And again, came from 3.5 million estimated cases yearly in the mid-1980s to 48 human cases, and a couple of thousand of let's say 2,000 of infected dogs. We have come from far. It's not a time to give up. There's room for understanding and pushing towards the last war in the world. The strategies, as we know, are still there, working, even though we need more operational research. But what we have as tools will lead us certainly to more successes if we use it very well. That means health surveillance and active case searches, detect and contain all infection, whatever it is, comprehensive vector control, cash reward, community awareness, social mobilization, access safe water supply. And this is what this is. Force, some images from the field. The cash reward, it's very important. So you have more eyes looking for guinea worm, not only us, more eyes everywhere, because people need to be motivated to do that. All countries have a cash reward system, very important, and now we need to advertise it, and it's being advertised actually. So, here is awareness creation. This is in Sudan, this is in Arabic. Sometimes I can read Arabic. Makoy can help. And then, this is how do we overcome the challenges. And you have, apart from the accessibility issue that everybody knows, we have a dog here, and not only dog, but animal infection. But this is being addressed by operational research to have better understanding of the transmission dynamics, and also to provide the tools, necessary tools, like CDC is doing right now, a serological test, and Georgia, University of Georgia, and Exeter University doing LAMP tests to see environmental sampling. How do we get more tools to really come up one day with, we're sure there's nothing left in turn to certify a country? Then Sharon and team was here. Thank you for keeping this one up, and we're getting there. So other challenges are the security, insecurity challenges. Makoy alluded at this a little bit, if not a lot, but South Sudan has the example of working, even weaving constraint, weaving insecurity, and to come up with results. Certification is the same thing. Nigeria, in 2013, when Nigeria was certified, we know Nigeria was difficult to be accessed. But the ICT team used the Red Cross and other NGO to access the area and come with enough evidence to say that, oh, there's nothing left there. So this is something that that can be done. But security is a problem. In Mali, we have problems in security. South Sudan is still there. And also CAR, the question that our colleague asked, CAR, what is CAR? So difficult to access. This will be least to say. So this is example. And sometimes ICT team, they have to be, accompanied by escort, military escort to do the work. And we have, for example, Niger and Nigeria and everywhere they have to be escorted when security is a concern. Soldiers escort. So I will share one small video of 45 seconds, advocacy video, to say that we can do it all together. What? There's no sound. The disaster. Okay. So, don't know if take this, and it's fine. But this is to show you that despite the last man, oh, good. So, South Sudan. I would like to thank the Carter Center, particularly Carter Center, for the dedicated commitment, and President Carter himself. I want you to applaud, because this guy have, for all his life, worked for a lot for us. And all the partners, including CDC and all the people who are doing a lot of research to help the program, and all the field workers. Makoy, you're here, part of the field workers. Please give a big applause to everybody who is involved. Thank you. Thank you.
Thank you, Dr. Sankara, for your presentation. I'd now like to call all the presenters up to the front. Hi, we do have about 15 minutes, and we welcome any questions that anyone might have, whether specific or general in nature.
Frank Richards, Carter Center. Thanks so much for the presentations and for the seminar and for the really wide scope of things that we've talked about today. My question is on the genetic part, very rich genetic part. Thanks so much for that. I'm still trying to sort out something in terms of, you showed us some markers that are the slower mitochondria markers, and then the other markers that are more likely to turn around quickly within a given area. So when you have a dog with multiple infections, this is what I didn't quite follow. Were you saying that the worms coming from that dog, multiple infections, were of the same lineage, or were the result, because you were saying something about the same maternal lineage, but that's the mitochondria. So what I'm trying to, did they have different fathers? Or, I mean, I'm just trying to figure out if there was like one worm release event that could have given multiple infections to that dog, or whether there is a series of infections. Obviously, this dog we heard about with 70-odd worms were likely a series of infections, would have had a lot of genetic diversity, but you probably didn't look at that. So is this a series of infections that the dog's being exposed to from different worms, is my question. Thank you.
So I actually did look at a subset of worms from that dog, and we do see a mix. So there's a mix of mitochondrial haplotypes suggesting that the dog was exposed to multiple different strains, multiple different sources of infection. And that has generally held out to varying proportions in other instances of multiple worms in a single individual, where we'll see some mix of multiple different haplotypes, but some repeated. So you kind of see a mix of potentially clustered transmission, but also kind of sampling throughout the environment. But I will say that on our initial analysis of the population, finding full SIB clusters, which would indicate transmission from one individual, like a cohort from one individual worm, that was actually fairly rare, at least within the dogs. So that would suggest that there were a lot of uncontained cases, as opposed to kind of a suite of individual cases that were responsible for infecting a large number of dogs. Conversely, that's kind of been the opposite with what we've seen with some of the human outbreaks. I mean, we've seen individual humans, but like the Salamat and the Ethiopian outbreak, we're seeing kind of evidence of true point source outbreak in those worms, or in those particular instances, which I think is also perhaps a fairly stark reminder that it really only takes one worm and one uncontained worm to cause problems for a fair number of people. And so we should really keep at this.
Hello, Justin Lana, Clinton Health Access Initiative and former Guinea Worm Warrior, 2013. I have two questions regarding Angola. I don't know much about population genetics, but we had the two samples. Just curious how many more samples you would need to find, or if there's a way to estimate perhaps potential population sizes with what's going on there. And then to the rest of the panel, just if you could give your thoughts on what we think actually is going on in Angola, what's the level of concern? Thank you.
Yeah, sample size is always, I think, a big question, and off the top of my head, I don't have a great answer. Unfortunately, this is one of those situations where not having a surveillance system in place is a detriment, because the condition of some of the worms is quite poor, so we don't get good nuclear data, like we're not getting good microsatellites from those worms, so that keeps us from being able to do kinship analyses that would connect the dots. So for example, one of the human worm from 2018 and the dog worm from 2019 currently appear to share the same mitochondrial haplotype. But that human worm, because it was so unexpected and there was no collection protocol in place, is deteriorated to the point where it's so far been very hard to get microsatellite data. So we may not even be able to do the kinship analyses that would allow us to directly connect transmission. I will say that at this point, even though they seem to be related to samples in Chad, I think the scenario that would have to play out for two different haplotypes that's closely related to make it into Angola is kind of a diminishing probability. And so it does seem like there may be some small scale transmission in Angola, particularly given the geographic distance between two of the cases that don't have any history of like long distance or international travel. So there may very well be some local transmission, although at this point we would need more samples to be able to determine if there's a large population size. No.
Do you want to talk about Angola? I would also just add that for Angola, Liz mentioned this, there is no surveillance system for guinea worm, and that's crippling from understanding whether there are samples out there to be collected, but also we have no sense of how dispersed guinea worm could be in the environment there. That being said, the National Ministry of Health has trained their health workers on guinea worm, and that's actually what led to the initial case being detected in the first place. They just happened to train a health worker who also owned a pharmacy, and that pharmacy, the small girl reported, or her mother or aunt reported her to that pharmacy. They didn't know what it was, they didn't know what to do about it, but through the training as a part of that National Immunization Day actually led to that. And so the Carter Center, WHO, working together to try and help build up surveillance structures in that area, though there have been some administrative delays to that process. But in the meantime, a number of investigations have been carried out, including by international guinea worm experts. And there's really no strong understanding by human populations about the existence of guinea worm in Angola. For example, I was there in August of last year, and we interviewed a number of elders and chiefs from the southern belt line of Angola. None of them were aware of guinea worm in recent history or even long-term history. There was one anecdotal report from one of them that suggested maybe there was a dog some years ago that might have had guinea worm. But again, we know dogs are afflicted by a number of different parasites and diseases, and so whether it was a migrant fly larva or something else, we simply don't know, and we have no evidence to corroborate that report.
This is a very good question indeed. Angola is a good headache to us, and this has been the case we detected because Angola had to fulfill the criteria of certification, then case searches and training, as Adam said. But currently, as per current knowledge, there is no evidence of a big, foci. The evidence, everything points towards a small transmission foci there. Exactly the extent of the area is not well, delimited so far, but we don't expect a full-blown, outbreak in Angola because we did case searches, interviewed people, and if it had been a big number of cases there, we would have found it now. So that is a small foci, but Ernesto came up with Ernesto Tiven Ruiz, that Dr. Ernesto that he went back and dig it out, and he found an article of guinea worm described in Angola in the 18th, 19th centuries. So this pointed out that probably it was there. It's not probably, but evidently it was there. But because the country was on a situation of war, civil unrest, up to 2002, So we are left over because nobody can go there and assess at the beginning when the program in 1980s was being established. Thank you.
Any other questions? Dr. Richards?
Sorry.
Provocateur?
Sorry. None for hogging the microphone.
No,
sorry.
you should.
I'm kind of, well, two things for coming back to the epidemiological presentation and your interest in looking south of another area, further south of the Chari River, where you felt that the features might be permissive for additional cases, and I wasn't really sure why you felt that way and what kind of approaches you might be taking. I mean, I'm really interested in the development of the serologic test, which you mentioned, and what the idea is for using that. I have ideas like, for example, in the Angola workup, you could go and test people sort of like a reactive case detection and test people there, maybe apply it in the area that you're mentioning in the south of the Chiari, or even if anyone could get into CAR and do a quick blood spot survey or something to be able to have a look. But I'm really kind of interested in how you're going to work that area up and how a blood test might be deployed. Thank you.
Thanks for your question. And a good question it is indeed. So for the first part of your question, you asked about what are the features or characteristics of that southern part of Chad and why is it similar to the Chari River. That area, which sort of surrounds the Lagone River, also experiences a similar degree of flooding and then subsequent formulation of oxbow lakes in that region. To my knowledge, people are also very much reliant on fish as a source of income, but also food. So those are the characteristics through my eyes that I see are similar. I'll also say that some of the very early human cases detected in Chad in 2010 were also in that vicinity. So I think it's certainly on our radar to begin active surveillance in that region. I agree that serology could be a very powerful tool. We are currently in discussions and using the serologic test in Angola to get a better sense of what seroprevalence is in dogs in that region. But I also think that there's utility for that in Chad in places where we don't have active surveillance, as you pointed out. So I think that utilizing the risk mapping approach together with the serological tool could be really powerful. So the risk mapping that we'll do will then point us to areas of possible high risk from an ecological standpoint where transmission could be supported, and then we could then go into those communities and conduct the serological testing for the dogs to see what the situation is, and then from there make a decision about the degree of active surveillance that we'd like to implement in those places. And I think that's the long-term plan, but there are many steps to get to that. But thanks for your question and comment.
Any last-minute burning questions? I'd really like to express my gratitude to all of the presenters today. Very much appreciate not only your contributions to the eradication effort, but your participation here today, and certainly to everyone in the audience, fellow guinea worm warrior or not. This has been a bold initiative from day one. In 1980, at the U.S. Centers for Disease Control, when they put this on the table as something to pursue, all the way to today, it still remains a very bold initiative, and we've come extremely far. It's that 0.1% at the end. There's a reason why everyone is talking about reaching the last mile, whether it's an elimination campaign or an eradication campaign. It's always the most difficult, requires a lot of patience and perseverance, and a lot of technical insight that we're trying to employ. And so we appreciate your being with us this morning, and thank you very much.
Would like to also thank you, and just take it where Adam left it. It's with time where we need more resolve than anything else. Because when it's like last mile is threshing long, people start to get discouraged, and some will say, "Oh, we cannot do that," more because we have dogs and animal hosts involved. But I will want to remember all of us that even rinderpest was not only was not even a zoonosis. It was a disease in animals, domestic and wild animals. But rinderpest was and is actually eradicated worldwide, let alone a zoonosis. That means that we have to keep faith and pushing. We'll get there. We get there because there is no evidence so far to say that we cannot do it. And actually, all evidence so far point that we can do it. So thank you for being here, and we will soon celebrate to eradication of Guinea disease. Thank you again.
It's a pleasure to meet you. It's a small world. Yeah.